Alright, you guys, here we go. Let me hurry and just swap this so it looks a little bit better. Alright, so you guys, today we're going to be going into gene regulation and expression, both in prokaryotes and in eukaryotic cells. And to just give you guys some extra information if you need to, this is in chapter 14, section 3 of the textbook that you guys have access to online. So if you need to read some additional stuff, I will also link some additional things below with this assignment where you can get some added information, because I'm just going to briefly go over this. This is it can be very complex, and I'm trying not to make it that way. And so if you guys need help with anything, send me an email or send me a message in Schoology, and I can even go into additional information. But I'm hoping that this quick little lecture and the additional information and sources that I'll put up will help you to better understand this stuff. Alright, so you guys, your discussion that you guys just did before kind of briefly went over this. I had you guys discuss, if you walked into a library and you saw all these different books that had all this information about how to build something or how to fix this or how to do that, the question is, would you need to use all of this information all at once? And obviously the answer is no. Sometimes maybe you're working out in the garden, maybe sometimes you're fixing a fence, or sometimes you're reading a book or something, and all that stuff. So you're using different information at different times. Our cells also are like this. They have to regulate. In our gene, every single one of our cells has all the genes to do everything that our body needs to do. But what we notice is that some parts of our body don't need that kind of information. For example, our skin cells are not going to express the same types of genes that our muscle cells are. Muscle needs to contract and help with movement, while skin does not do that. It helps to protect our body. It's our first defense against our immune system, or for our immune system, against bacteria and viruses and other pathogens. So each part of our body, every cell is different and has to do different things. So the question is, how does it regulate the genes that it needs to express? And we're going to dive into that today. To get started, though, I have a quick little video that I want to show you guys, kind of like phenomena to kind of look at. Let me go to where we need to be here.
On the gene misfired, flies grew legs in the wrong place, on their heads in place of antennae. In normal flies, legs grow from the midsection, the area called the thorax. So Levine and McGinnis decided to hunt for the gene in the thorax of a normal embryo.
The expectation is that antennapedia would be active, expressed in the thorax, the developing thorax of the embryo. But who knew?
Levine and McGinnis had to...
Okay. So you guys, kind of an interesting aspect that I want to go over real fast for you. So what they kind of discovered with this is that, here, I'll go right there. No.
As you looked at these genes, you said this one.
Let's see. Okay, right here. Alright, so what they kind of did, you guys, scientists have discovered that our bodies are made up in sections, and each section is coded by a different gene, okay, during early development. And what they started doing is, okay, well, what is making this section different than this section? And they were kind of looking at that, and so they were starting to practice, well, what if I had the cells in here that are normally expressing a particular gene? What if we had the same thing happening right here? And so they had so they were doing some tests and things, and they were able to find that this gene that codes for maybe these legs are turned off in the head region. Antennae are a little different, right? And so they were kind of looking at that, but kind of a fascinating thing. What the heck is going on there? Why is this fly growing? Where's that? Growing head or legs out of its head, where the antenna should be. So we're going to really look at this and see, well, how do cells regulate the different genes that are being expressed there? Okay. All right, so to get started, you guys, we're going to look at prokaryotic cells first because they're a lot more simple than eukaryotic cells, and this is actually where we first started to better understand how genes get regulated. And there's a couple quick things that I want to go over. First thing is just know that that genes can be controlled. And they can control that transcription in those genes. And remember, just to refer back to protein synthesis, we have transcription and translation. Transcription is where the DNA is making a copy of itself into RNA, and then that RNA will leave the nucleus or go to the ribosome, wherever it might be, and get translated into a protein. And so what we've actually found is that cells, both prokaryotes and eukaryotes, can regulate transcription. And in bacteria and other prokaryotes like this, what they've actually seen is operons. And what we know about operons are groups of genes put together that can be turned on and turned off in those cells. And so we're going to dive into this a little bit here. So let's dive. So first of all, just know that the lac operon is the one we're going to really look at here for this example. Now, the lac operon is specific. If I remember right, it's made of 3 different genes, and these 3 genes get turned on and turned off in certain scenarios. And what they do is they help when there is lactose present. Now, what lactose is? It's a milk sugar. So whenever you drink milk, there's lactose. That's the type of sugar that's in that. And that's why people that are lactose intolerant cannot drink milk, is because basically their bodies cannot break down this type of sugar, lactose. Well, bacteria, they're very helpful, and we actually have, this is E. coli is what this is showing. E. coli is in our stomachs all the time. E. coli help us to break down this lactose. And so, but when bacteria, they don't always express those lactose genes. If there isn't lactose to break down, they're not going to express those genes, if that makes sense. And so they'll turn those lactose gene, this lac operon on when it needs to. And so we're going to dive in to see how it kind of does that. So right here is kind of a little figure that shows this. When we're looking at the lac operon and all operons in bacteria, and in prokaryotes, we see this kind of thing. So right here are the lac genes, the 3 lac genes that would be turned on when lactose is present. We see that there's a section of before upstream of these 3 genes called the lac repressor gene. We also have a major point called the promoter, and then we have what's called the operator, and I'm going to kind of show you what these things do here. But basically, the promoter is a place where RNA polymerase will bind. Now, just a quick refresher, what does RNA polymerase do? Well, it's going to hop on, and it's going to skim through this and make a copy of RNA of that gene, of these 3 genes. And so as it's going through, it would pop out an RNA for that, pop out an RNA for that, pop out an RNA for that one to go and be translated at the ribosome. The operator, this is where this lac repressor gene, if it gets coded, it's going to make this active repressor. The active repressor will bind onto the operator. And what this kind of does, you guys, is if this is binded to the operator, if you notice, RNA polymerase, when it binds to the promoter, it can't go. This blocks its way. And so this is one way that the cell, the prokaryotes, will stop the expression of these 3 genes, is by having this repressor binded to that. And as long as it's binded to the operator, these 3 genes cannot be expressed. Yep, went over it. Thank you. All right, so the lac repressor blocks transcription, kind of like what I talked about. So when RNA polymerase, it cannot bind to the promoter, and it cannot transcribe those 3 genes. So what does the cell do to get rid of that when it does need to present that, when it needs to express those 3 genes? And so we're going to look and see how this kind of works. So when lactose is present in that cell, so when, say, for example, the bacteria are living in my little stomach, I love milk, drink it all the time. So I take some milk, I drink it, and now it's in my stomach. Lactose is now present. If lactose binds to that repressor protein, it will actually release it from the operator, Okay, and so that allows RNA polymerase to bind to the promoter and run downstream and create that mRNA from those proteins. Okay, and from that we get 3 different proteins that get produced during translation. Okay, so just a quick little refresher here. When it's being shut down, it's when what's not there? Lactose. So lactose is missing in that cell. It does not have it to take off that repressor protein. But when there is lactose present, that will get released, and RNA polymerase will then say, okay, let's make this gene, let's make that mRNA get translated. Okay, so quickly, like what I said, lactose will turn it on when it is present, and when it's not present, it will turn it off. Okay, now they found that to be how it kind of works in all cells, except it's not always lactose that's there, right? It could be a ton of different things that would turn on and turn off those operons, those groups of cells. And so when we're looking at this, let's dive into a more complex aspect of this, the eukaryotic gene regulation. Now, you guys, eukaryotic gene regulation, it's a lot more complex, and we're going to go over this in a very basic and simplified manner. It is not this simple, and even in prokaryotes, it's not as simple as I stated. But for for just the depth that I need you guys to know, this is what we're going to go at. But just know that eukaryotic gene regulation is a lot more complex. So when we're looking at eukaryotic gene regulation, there's some different parts here. First, we have what are called transcription factors that can bind to these enhancer genes that will help to turn on or turn off those different things. The TATA box is kind of that place where it starts it all out. Okay, and what it is, it's they would actually see TATA. Or TATAAA. They call them the TATA box. But it's a pattern that kind of tells the RNA polymerase, hey, get in there. That's where you need to start. Okay? And when it does that, so you can see that the enhancer proteins will bind to those those transcription factors, will bind to the enhancer gene, and then will cause RNA polymerase to transcribe in that one direction. But you see RNA polymerase will bind to that TATA box. Okay. So by binding DNA sequences in the regulatory regions of eukaryotic genes, transcription factors control the expression of those genes, kind of like what I said. So it's these transcription factors that really start this all out and drive that through. One thing to note that I want to just say here really fast is that understand that gene regulation in eukaryotic cells can be regulated at a number of levels here, right? And so it's kind of interesting to see how the cells can kind of regulate and turn on this gene. If they need to have this gene expressed in a lot more detail, they could cause that to occur by these transcription factors. Let's see. I just want to make sure I've covered everything that I wanted to cover for you guys. Yeah. Okay. So I think we're good there. I think I covered everything. So just the number of transcription factors that are present here can speed that up or slow it down, which is very interesting and complex at the same time. Anyways. All right. So cell specialization. So kind of like what I talked about, we have skin cells, we have muscle cells, we have nerve cells, all these different cells that do different things. How is it that the cell will regulate those different different cells to make them unique and specialized? And that's how it kind of does that here, is using these regulation type of proteins or regulation processes. So it'll turn on certain genes, turn off some genes that don't need to be expressed. And so going back to the phenomena that I started with, when it comes to the Hox genes, this kind of shows that, right? And so here are different types of tissues throughout the body as development occurs that will need to become specialized. And so looking at that, you can see the genes that are being turned on and turned off. And what they've actually noticed is that all organisms follow this same pattern where some genes are being turned on in certain parts of the body during early development that help it to develop these different body parts that it needs, which is very fascinating. And when we look at evolution, this is a piece of evidence that shows all living things are related and came from a common ancestor. So homeotic genes regulate organ development during early development of the organism. Yep. Yep. Talked about it. Okay. So this was actually where I did a lot of my master's degree was with epigenetics, and I wish we could spend a lot of time on here, but I'm just going to briefly go over this. And if you are interested in this, hopefully we can go back to school and have a normal life again. And if that's the case, take my genetics class because we spend some time on this. I really think it's just such a fascinating topic. So you guys, epigenetics is another aspect and way that our cells and our body can regulate different genes, either by turning them on or turning them off. And what we've actually seen is basically epigenetics is how the environment impacts our gene expression. So we've been going over and looking at how just in our side of our cells, how it can turn on and turn off genes normally. But sometimes what we get expressed or exposed to in our environment can impact this. So they've actually found that a monarch butterfly it goes through development and it's being pushed by what we call epigenetics. It controls those genes that are being turned on and turned off during each phase of that of the insect life cycle, depending on the environment indicators that it's getting. And so here's another example of this. So environmental influences. Here's a rabbit. If the temperature is dropped below 25 degrees Celsius, it'll cause some fur to go black. The question, I wonder why, right? There's a couple of theories that I have. And just so you guys remember, when it comes to genetics specifically of this is Himalayan, so it's a type of multiple alleles genetic problem. But we can see that just because they're Himalayan, temperature can cause some unique aspects to their fur color. Okay, but back to what I was saying. When I look at this, I see these as extremities, right? And so we see their nose. What gets cold really fast when you're outside? Well, your nose, your ears would, and then your feet and tail. Well, black will attract the sunlight and maybe keep that warm. And so I'm wondering if that is why that is happening there. And so the environment is pushing for these genes to change color when temperatures drop below that. And so if the bunny is living in these colder climates, it has actually a higher chance of not getting hypothermia in those locations and losing those extremities. So that's my theory. All right, so to end off, you guys, I want to play this quick little thing. And this is current news. I've talked about it sometimes with you guys, but I want to quickly play this. So basically, you guys, what this is, now that we understand a little bit more about how genes can be expressed, turned on and turned off, there's been a lot of research on this. Because the question is, how can we then cure some cancers or cure some diseases? If we now understand how we can turn on and turn off certain genes, could we then turn off some genes that cause, for example, Huntington's disease? Huntington's disease is a dominantly inherited trait, and basically, if you have one dominant allele of that, you would then express it. You don't know you have it until later on in life. About in your 50s or 60s, you'll start to express Huntington's disease. And basically what it is degeneration of your nerve cells in your brain. And so you lose functionality. It's very hard on them and on their families. And so the question is, could we then use this and say, okay, well, whatever gene is turning that on later on in life, could we shut down that gene that is causing that to occur? So let's watch this, and it kind of goes briefly over how this new process called CRISPR-Cas9 kind of works.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces 2 types of short RNA, one of which contains a sequence that matches that of the invading virus. These 2 RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use 2 tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
Okay, so you guys, I think that is a very fascinating thing, and I'll put actually another link to this assignment as well that kind of goes over some moral implications that come because of CRISPR-Cas9 as well. Anyways, there will be a quick assignment that I want you to do using CRISPR because I think it is such a fascinating opportunity and thing that we've got coming up that could revolutionize medicine as we know it. Anyways, you guys, if you have any questions, that is all I was going to go over today. If there will be a few quick little questions that I want you to answer. Again, if you need to use the textbook or my other resource that I have on here, please do so. All right, you guys, I'll talk to you guys later.